This is DNA part 2. Some of these bits of information we've actually talked about before, and so will be very familiar to you and easy, I hope. Others you might be familiar with from any experience with forensics, and there may be just a couple of new definitions for you to get under your belt, so this shouldn't take too long. First off, we're kind of getting into DNA and how we use it in technology these days. First off is an old idea. Selective breeding, which we've learned about before, is the process of selecting organisms with desired traits to reproduce in the hopes of having more babies with those desired traits. What you see in the picture is what was done over thousands of years with corn in the New World by native people. They took this guy here, which is sort of wild corn, and kept selecting for the ears that had the most kernels until eventually we got to what we currently market today and grow as corn. We've also done selective breeding with the wild mustard plant. This guy here has turned into cauliflower, broccoli, cabbage, kale for us over many generations. Of course, we've already talked about how dogs are impacted by selective breeding, but you can also see it affects corn. There's all sorts of options of things that one can do. So this leads us to genetic engineering, which is just the next sort of step along the way from selective breeding. What happened was that when scientists discovered DNA, they found new methods because this provided a new way of sort of getting desired traits into organisms. Because now we are no longer limited by the same species to some degree, because we can take genes from one organism and actually insert them into the DNA of another organism in order to transfer whatever ability that gene had, whatever protein that was being made by it, whatever production of a pigment, whatever production of a, maybe a viral spike protein or something like that. Genetic engineering allows us to do that. Okay, just moving genes from one organism into another in order to transfer some sort of ability or be able to produce a particular trait. This is the way that we sort of used to more traditionally make vaccines, or this is one of the ways they traditionally used to make vaccines. It just involved getting, for instance, in this example, we have 2 flu strains. Flu 1 is the one that's causing problems. Flu 2 is the one that doesn't really impact us. It doesn't cause any problems in humans. And so they're trying to transfer some of the surface protein genes from flu 1 into flu 2 in order to make a vaccine that can then be given to people. And the flu strain 2 doesn't harm people, but it'll allow them to recognize those problem proteins on the flu strain 1 and thereby hopefully give them immunity to it. So they just take the genes out, they splice them out of the DNA that's inside of the virus, they put them into these plasmids, which are these circular pieces of DNA. Each of those plasmids has a different component for flu strain 2. And then they just added the proteins from flu strain 1 that are on the outside that don't cause any problems. They've eliminated all the harmful genes that were in flu strain 1. And then they insert that and grow them until they get enough of the virus produced by these, by inside these animal cells, because they grow them in usually animal cells in the lab. And then they harvest out those viruses, those flu strain 2 viruses that now have the protein from flu strain 1, and they could use that in the vaccine to give to people. DNA or fingerprinting is another thing that some of you may have heard of watching crime shows. Human DNA is very unique from person to person. but it's the same in all of our cells. So every single one of our cells has the exact same sort of DNA in it, except for maybe a few skin cells here and there that may have mutated. A technique called DNA fingerprinting produces an image of patterns made by cutting up your DNA and magnify, like making a whole bunch of copies of the DNA, cutting them all, making them all cut into different sizes, and then those sizes are run through a machine that spreads them out according to their size. And then a picture can be taken of that, and that picture is unique to you and can be compared against DNA samples that they may have retrieved elsewhere, possibly at a crime scene, to see whether you were the one that did it. That's a very simplified way of what's going on here, but there's a little bit more on the bottom here if you wanted to read, but it does, that's basically it. You take out the DNA, you cut it up, you allow them to make copies of all those cut pieces, then you separate them into like this little agar plate that allows different sized pieces to move at different rates through it. And so they all spread out at a particular pattern, and that pattern, take a picture, that's your DNA fingerprint. And they are unique to you. So they can be used in also paternity cases or maternity cases, although maternity is usually not as big of an issue. But you can see in this example, there's a mother and a child, and then there's potential parents 1, 2, or 3. And you can pick out, hopefully relatively easily, which one seems to be the most likely parent, I would say. Yes. All right, the human genome, another definition for you. Probably heard the word, maybe don't know exactly what it is, but a genome is the total amount of hereditary material in a single cell of an organism. So put it all together, that's your genome. They're, again, individual to each individual person. Fun fact, if you spoke 1 letter of DNA per second, 24 hours a day, it would take you 100 years to go through the entire human genome. Mitochondrial DNA is something else that is considered part of the human genome. It's the DNA that is found inside the mitochondria of a cell. It traditionally is passed from mothers to children. It doesn't come from the father at all. So it's another way that we can look at how to trace human origins back, because if we follow the changes to the mitochondrial DNA back through just the mothers, it can tell us quite a bit of information because it's a direct ancestral line. Pretty cool. Problems with mitochondrial DNA can also cause major problems in people that can cause diseases. Other fun facts about our genome. Of all the genes in the human genome, 55% were already present in the first evolutionary animal. And that's what I mean by kingdom Animalia. So the first evolutionary animal, maybe some sort of sponge or something, 55% of our genes were in that first animal. Pretty cool. Humans and sharks make hemoglobin for their blood using nearly identical genes. It's almost an identical process. Same gene, same hemoglobin, same purpose. That's kind of cool too. You even have something in common with single-celled protozoans. So protists, kingdom Protista, they have some genes to make proteins that let them cling together in tiny colonies. In animals, those same genes help cells glue themselves together in order to build tissues and ultimately form your multicellular organism body, which is kind of cool. And scientists estimate that humans contain between 20,000 and 25,000 genes. There are thousands and thousands of genes in your DNA. We don't even know what all of them do. And then that's basically it. We are going to be doing a little bit of a DNA forensics activity in class. Sir Alec Jeffreys is the guy who came up with this technique of DNA fingerprinting that we now use in forensics today. And it can be used to solve a lot of different crimes, and not just with humans, as you will find out in our lab. Okay, and that'll be it.